The eLife Podcast from eLife, the open access journal for outstanding research in the life and biomedical sciences. Online at elifesciences.org.
Hello, welcome to episode 39 of the eLife Podcast, which is produced by The Naked Scientists. I'm Chris Smith. This month, TB, malaria and schistosomiasis. Is there a connection? Well, the answer is they're all neglected tropical diseases, one way or another, which is what we're focusing on in this episode of the programme. But why are they neglected, and why shouldn't they be? From the University of Toronto, eLife Senior Editor and Professor of Global Health, Prabhat Jha.
When we talk about neglected tropical diseases, they represent a grab bag of actually quite different parasitic or vector diseases which actually are quite different in different settings. African trypanosomiasis, schistosomiasis, leishmaniasis, leprosy, dengue, rabies, things like hookworm disease. What is common to them, they actually don't kill in very large numbers, but they cause a substantial amount of disability. The second thing that unites these diseases, they are among the most heavily concentrated in the poorest parts of the world. Now that's important because getting rid of them has double challenges. You're working in difficult places of the world. The upside is control of these diseases is probably one of the best pro-poor things the world could do in poor countries.
Really, the spin-off of what you're saying is if we intervene in these countries, A, the money goes further because they're poorer countries and things tend to be a bit cheaper, notwithstanding the challenges, but B, there's a huge economic benefit because of the way these diseases cause chronic disability.
Absolutely. Most of these conditions, take leprosy, which is still quite common in India, is a big cause of poverty of the household. Visceral leishmaniasis, which again is in many parts of India, causes lifelong disability in those affected. These are eminently treatable, and we have had successes. We have been able to get rid of guinea worm in Africa. Yaws is on its way to being eradicated. And there are targets. The World Health Organization has set targets for getting rid of leprosy, trachoma, and a few of these other conditions. It's going to be difficult because, as we know from the experience in eliminating polio, when you start out in a campaign to get rid of these diseases, the first few cases you detect and treat are generally easier to find and cheaper. But as you go along, they become harder to detect, and therefore they need more resources, and they need newer science as well.
It's also a moving target, though, isn't it? Because with polio and things like that, we have seen cultural barriers to eradication. We've seen wars.
Absolutely.
So we've seen a lot of human factors intervening, which actually have thrown a huge spanner in the works, and we would probably be a lot further along. And it wasn't the science, it wasn't actually the funding that held the process up, it was other people.
Absolutely. And those are part of the challenges of working in these poor countries. But the good news, I think, is the world made progress on polio because it put money into it and it invested in moving from oral polio to injected polio. And there was good scientific thinking behind that because the injected polio was less likely to cause disease in a way that oral polio would. And we know that even earlier on from smallpox eradication, that it required a new technology, which was basically using ring vaccination, finding a case of smallpox and vaccinating everyone around that. That scientific breakthrough of using ring vaccination was exactly what the world has tested in the latest Ebola vaccine. So the key there is that as these targets get met, it will need a lot more money, and the last mile is always the more expensive.
Does it not come down to human population? We're invading more virgin territory where these diseases are endemic in nature, and it's more likely we're going to see spillover into humans. So until we get to the bottom of the population problem, we're really ignoring the elephant in the room.
Well, I think they're related, but not causally related. But the positive news is that if you look at areas where there is an increasing urban concentration, and you've got urban infrastructure, you can actually have clinics, then it is in some ways easier to control these.
Well,
Now we
ironically,
have,
people said that with Ebola, it was people coming across the border to come to these towns where there were, albeit not terribly good, but there were medical facilities, and this was actually helping to spread the outbreak.
That's right. And the other factors which is important in Ebola was the burial practices where you had to hug the body goodbye. The response to it was late, but very crude isolation strategies that were brought in were effective.
So we need better resources. We need more science thrown at this in order
Yes.
to be more crafty and clever as time goes on. But will people pay for it? Because the harsh reality is Ebola, the vaccines, we now have something like 7 different vaccines in trials at various stages of development or more for Ebola. Now, they're not rocket science. Those vaccines have been made using fairly mundane techniques, actually, that we've had for decades in some cases, yet no one wanted to make them and deploy them because they didn't think
Yes.
Ebola was a country's problem that had the capacity to make those vaccines.
In the short or medium term, it's in everybody's interest to stamp out epidemics where they occur because it spreads. But I would argue that public funding of science, which has really decreased since the 1970s, really needs to be back on the same level because that science creates products and tools that you can use to fight epidemics. The private sector won't take care of these epidemics. They have no incentive to.
So politicians take note. Prabhat Jha there from the University of Toronto. One of the newest kids on the biological block is Zika virus, which we know to be spread mainly by members of the Aedes family of mosquitoes. But that doesn't mean that other species can't transmit it too. But which one should we worry about? To find out, Michelle Evans and Courtney Murdoch from the University of Georgia have taken a leaf out of the way that popular music gets sold and marketed. Courtney.
This was an attempt to try to be proactive. And so we developed models that allowed us to make a list of potential mosquito species that could be important transmitters of viruses like Zika for a given area, for the United States, for example. As an empiricist, it's really nice to have a list of species as well as their potential rank order of importance so that we can streamline empirical efforts to look more closely at these species and their relative importance for transmission.
And Michelle, how did you approach that? Because there are many, many different species of mosquito, and they have many, many different geographies in which they can operate. So how did you do this project?
That's definitely correct. There's over 3,000 species of mosquitoes in the world, and it would just be impossible to empirically test them all. And even to create a model that could consider all of them would be hard. So we kind of focused on a subset of the mosquito species that transmit a certain group of viruses that are very closely related to the Zika virus. We created a model, kind of like a recommendation system that Spotify or Pandora might use to recommend a new musical artist. And so the way these systems work is based on the traits of artists you like. Their algorithms will recommend new music based on a ranking system. Our model does the same thing, except in place of an artist's genre or the rhythm of the music, we used a common mosquito and virus traits, such as their geographic range or disease symptoms or severity. And then this allowed us to get a list of mosquito species that could potentially transmit Zika.
Now, what geographies were you considering, or were you looking everywhere?
So the way we used that trait was more about the presence of a species in a certain area of the world. We had a couple different traits. One was how widespread is the species, and another is if it was specifically on a certain continent.
Right, and you marry that with the likelihood that there is a virus a bit like Zika being circulated in that area and how much.
Yeah, and then also the presence of other mosquito species there that might carry the virus. So I was able to draw on these connections.
And Courtney, when you do this sort of analysis, what did you actually see?
What was produced was a list of potential mosquito species in the U.S. that could be potential transmitters of Zika virus and other viruses similar to Zika. And this was rank ordered. So obviously we had, the top 2 candidates were species that people would already expect, like Aedes aegypti, which is the yellow fever mosquito, which is the primary vector for dengue and chikungunya. as well as Zika. And then Aedes albopictus, which is the Asian tiger mosquito, which has a much wider distribution. But then there were some other candidate species that were slightly surprising. So Culex quinquefasciatus, a species that does live in close proximity with humans, but has been more involved in transmission of other viruses like West Nile virus, came up as a potential candidate. And we had some other Aedes species come up as potential candidates.
And Michelle, these data obviously are predictions, as Courtney says, about what mosquitoes might transmit Zika. But at the moment, presumably, we don't know. We just know that they might because they help to transmit viral relatives of Zika. Therefore, they might be involved. So is the next step then to actually go and test some of the leading candidates?
Yes, that's exactly right. In our paper, what we really strongly recommend is that this provides a list for empirical tests to be done, and that as these tests are being done, existing ideas about the biological realism of these mosquitoes is also brought into play. So, for example, some mosquitoes that we predict might transmit Zika might not really be in close proximity to humans, or they might not prefer to bite humans. And so those kinds of species can be discounted from the list relative to ones that we know are more likely to bite people.
Could you, Courtney, translate this to other geographies? You focused on the Americas at this stage, but would it work elsewhere?
Yes, I think our approach would work for any given geography.
Michelle, bottom line of this study, if you had to summarize in one sentence and say to politicians, this is what we found, what would you say?
I think the important kind of takeaway from our study is that this is a method that can be used, especially in future disease outbreaks or even before those outbreaks happen, to help get researchers on the right track as far as which species they should be considering, or even vaccine development, which are the viruses we should be targeting. And it just helps us prioritize where to spend our limited time and resources.
Michelle Evans, and before her, Courtney Murdoch. On Michelle's list are 35 mosquito species that might be able to transmit Zika virus. 7 of them are resident in the US, and together they cover the entire US mainland, so Zika might ultimately affect a much larger region of the United States than had previously been anticipated. We'll have to wait and see. You're listening to the eLife podcast with me, Chris Smith, and still to come, new insights into how TB escapes the clutches of the immune system and how stem cells help Bilharzia to stay one step ahead. Before that, though, in numbers terms, malaria is probably the world's most important disease. But its life cycle, moving from a mosquito's blood meal into the insect's salivary glands and then back into human blood via a liver cell, makes unpicking some of the aspects of malaria biology very difficult. Dennis Klug has been studying what goes on midway along the mosquito's intestine, where it forms a structure called an oocyst, where the infecting parasites multiply. And he's found a critical gene that releases the progeny into the mosquito's blood.
The parasite has to colonize first the midgut of the mosquito and develops into a stage that is called the oocyst, where it multiplies. And from these oocysts, parasites are released into the blood of the mosquito. And these parasites are called sporozoites. And these sporozoites then invade the salivary glands and can then be transmitted with the next bite to a new host. We are interested how the sporozoites are able to do that, egress from the oocyst and to invade the salivary glands. So what we do is we take infected mosquitoes and then we extract the salivary glands. Then we can extract these sporozoites and then we just image them. And these sporozoites are really highly motile cells and make continuous circles on this microscopy slide, which makes them very easy to image.
What about the oocyst, though? Because that's a different beast entirely, isn't it? The one that's sitting in the midgut of the mosquito and needs to get into the blood of the mosquito. It can't be very easy to see in there.
What we are especially interested in is the motility of these sporozoites. And we know that there are certain surface proteins called adhesins that play a crucial role in this interaction of the sporozoite with its environment. And we found a new protein that was not previously investigated. And by making a gene deletion of this protein, we found that the sporozoites are no longer able to egress from this oocyst, probably because of a motility defect that makes them unable to break free from this rigid membrane.
What's the protein, and how did you find it in the first place?
We believe the protein is a surface protein, and we found this protein because the adhesins that are described already in the literature, they have a very specific domain organization, and we looked then for proteins that have a similar domain organization, and we found this specific one, and then we investigated this one by different genetic approaches, tagging the protein with a fluorescent tag or by gene deletion.
So you knock it out, and the sporozoites can no longer escape from this oocyst. Have you done the experiment to put it back in and prove, just by discreetly putting that protein back in, you can restore the ability of the sporozoites to get where they need to go, thus proving it must be that protein that's responsible?
Yeah, exactly. We did 2 different gene knockouts. In the next step, we rescued the effect by putting the gene back. And once the gene is back in place, sporozoites are again able to egress from the oocyst and also to invade the salivary glands.
And do you have a feel for what these proteins do yet, or do you know that they're only involved in helping the sporozoites to move?
It's not known exactly what these proteins are actually doing. There are some speculations that some of these adhesins have adhesive properties. Parasites need them to adhere to substrates. It could also be that some of these proteins play a role as a sensor. The parasites needs them to interact with the environment and to sense where it is or if it is in close proximity to other parasites.
And are they used exclusively in the escape from the oocyst, or does the malaria parasite use these same genes or the same protein at any other point in its life cycle? Because it's got to go through a lot of transitions between different tissues and different cells in switching between the different hosts it infects, hasn't it?
The specific protein that I investigated in this study was also important for the invasion of the salivary glands, because we found that in some mosquitoes, some of these sporozoites are able to egress from the oocyst, probably because mechanical stress, and these sporozoites are then also not able to invade the salivary glands. And it is known that other adhesins play a role in salivary gland invasion, for example, in motility, but also in migration through the skin once these sporozoites are deposited in the skin of a bitten human, for example.
Dennis Klug, he's at the University of Heidelberg. It was in 1882 that Robert Koch announced to the world that he discovered the cause of the disease tuberculosis. He did it, in part, by looking down a microscope. But although Koch linked TB bacteria to the disease, despite more than a century of study since, we still don't really know how the infection unfolds in the average person. Now, Africa Health Research Institute scientist Alex Segal has been looking down the microscope himself, and he's got a new piece to add to the puzzle.
TB exposure is very common. 1/3 of individuals throughout the world are exposed, mostly in developing countries. 10 million active infections and about 2 million deaths a year. It's quite a healthcare burden, and it actually is a burden in countries which can least afford to deal with it.
The causative organism, it's a bacterium, a mycobacterium.
Yeah, so it's Mycobacterium tuberculosis, and it's been studied for about 100 years or more.
And what's our present understanding of how it causes the diseases that it does and how it spreads?
It's mostly a disease of the lung. It spreads by aerosol. So people cough, and somebody actually breathes in the droplets. Inside the droplets are the microorganisms.
And once they land in your lungs, what do they do?
They're taken up by a cell called a macrophage. At some point, that cell may attract additional cells, and this whole conglomeration of cells becomes a structure called a granuloma, where you have some infected macrophages at the core and an outer cuff of fibers and other immune cells.
The mycobacterium is actually inside the infected macrophage. It's not killed, it's in there and viable.
It's in there. In most cases, it's quiescent, and the person's immune system is able to control it.
And so what about that model were you uncomfortable with, which provoked you to start looking in the study that you're presently publishing?
Well, the transition to really the active disease is not that clear. I mean, it's known that the immune system controls it. But let's say now the immune system stops controlling it. What are the steps that need to happen for the infection to actually grow?
In other words, in the vast majority of people, the infection gets into the lung, but then the immune system holds it back and doesn't go anywhere. But in a number of people, and for various reasons, it suddenly becomes productive and it starts to grow in the infected cells, in the infected person. But we don't really understand what the triggers are or what the immune processes are that allow that escape.
Yeah, that allow not only the escape, but also the continuous growth of the bacteria.
So how did you manage to study it, given that people have been looking at TB for a century and they haven't yet been able to find this?
The tools that we use are fairly recent, so it's time-lapse microscopy, and we figured kind of the best way to understand it is just to see it, and we just filmed the infection. So we used a very simplified model, so we just infected macrophages, and we looked at what happens, how the infection actually grows or does not grow.
And what did you find?
We're actually surprised. First, that these cells are tough as nails, the person's cells which contain these mycobacteria. They can contain the infection when they're infected with low numbers. Up to about 50 bacteria, they're okay, but above that, they start to die. But what surprised us more is that the bacteria don't die with the cell, with the host cell, and they grow very nicely in the dead cell, where before, in the live cell, they were actually controlled, even at high numbers.
Oh, so basically by killing the cell, they've now released a whole bunch of nutrients and foodstuffs that those microbes can then assimilate and use to fuel the further amplification of numbers.
Yeah, so it seems. So the cell before was contained them in a compartment called a phagosome. Once the cell is dead, it's dead. It's like a sack of nutrients, whatever is left of it.
And then what happens?
Well, macrophages are phagocytes, so they pick up all the junk and bacteria and so on that surround the lung. But they will not engulf another live cell. But what they will do is engulf a dead cell, even if it's infected with a lot of bacteria. So you have the first cell which died because of a high bacterial load. It's going to be engulfed by another cell. Now that cell is faced with an equal or greater problem because now the bacteria has some chance to grow. Now, once that cell dies, the bacteria have extra nutrients, and the next cell is going to come along and pick that cell up. So basically, cells at that stage of infection kind of have two functions. One is they provide fuel for the infection to grow, and second, they're bait for other cells to come in and engulf them and die.
So in a nutshell, we have a sort of three-phase life cycle going on where you have an initial infection. If the number of bacteria in the initially infected cell increases to beyond a threshold of about 50, the cell is compromised. Yeah. In the course of dying, it then becomes bait to pull in more potential victims. And so you get this sort of positive feedback loop going on where you're amplifying the bacteria, feeding them in the process and attracting more food as they grow.
Right. The only thing you need to start this process is you need enough bacteria in one place. So the bacteria need to be concentrated in a clump. And this is in fact what the TB naturally do.
Alex Segal. Last year, we heard from Jim Collins at the University of Texas Southwestern Medical Center. He's been studying schistosomes, and when we last spoke, he'd just discovered one of the ways that these parasites, which multiply inside freshwater snails, then infect humans when they enter the water, fend off our immune response. I caught up with him to hear how the work's progressed.
Schistosomes, which are these parasitic flatworms that infect hundreds of millions of people, have stem cells, and once they're inside their human host, they're capable of surviving there for decades. And we've postulated that the stem cells are playing an important role in keeping their tissues young and healthy throughout this really long lifespan that they have.
Because one of the big challenges is these parasites have to live up close and personal with the immune system, and they're therefore potentially being attacked and assailed all the time, aren't they? So they may well have tissues that need replacing.
Exactly. So they live in the blood, which is basically the front lines of the immune system. And so what we found is that these schistosomes, these so-called blood flukes, they have these stem cells, but it seems that the main job of these stem cells in the parasite is to constantly rejuvenate the parasite's skin, which is a structure called the tegument. And so, it suggests that the stem cells are playing a role in perhaps allowing the parasite to maybe even to survive in the blood, but maybe also as a means that they are able to evade the host immunity.
Now, that's sort of where we left off when we spoke last time, but how have you taken the story forward and actually challenged that notion to say, well, if that's true, we'd better prove it?
So it turns out that that's a very challenging experiment to do, to directly test the model that the stem cells are involved in modulating how the parasite interfaces with the host. And so what we've been focusing on is the developmental aspects of how the stem cells are rejuvenating this tegumental structure.
Presumably, if you were to disable this system, then you would render these animals much more vulnerable to things like immune attack, and therefore the likelihood of them being cleared from the body would go up enormously.
Exactly. So if we could figure out a way to block the stem cells' ability to make these tegumental cells, then that absolutely could be a way to think about trying to target these parasites. If you blunt their ability to make new tegumental cells, our hypothesis is that then the parasites would no longer be able to survive the hostile environment within the immune system. And those are experiments that we're actively trying to get to work in the lab. It turns out it's not such an easy experiment because what it actually requires is a viable immune system, which we have really no means of replicating inside of a dish where we in cell culture where we do a lot of these experiments with the parasites. What it's going to require is us taking parasites out of a host such as a mouse, manipulating them to deplete their stem cells, and then transplanting them surgically back into an animal that's capable of evoking an immune response against the parasites. And so we've developed a procedure based on some classic literature to be able to do this in the lab. We've successfully done that. And so now we're working to basically kill the stem cells and then put the parasites back in and see what happens.
Is there no way you could wire some kind of genetic vulnerability into the cells so that you could, for instance, administer a drug and then kill them off that way?
Yeah, that's, it's one of the challenges of studying, a neglected tropical disease is that in many ways, the organisms that we use have been experimentally neglected, and we don't necessarily have the tools that you have to study things like fruit flies or nematodes. And so one, kind of pie in the sky idea that a number of people in the community are trying to develop is genome editing techniques, and where you basically use this system called CRISPR-Cas9 to make changes in the genome of the organism, and then you can exploit those to try to understand the biology of the animal. Unfortunately, given the way the parasites live, they have a very complicated life cycle. It makes it really challenging to try to develop ways to implement these kind of new genome editing technologies to manipulate these parasites.
Jim Collins, and on that note, that is it for this month. You've been listening to the eLife podcast, previous editions of which are also available on eLife's website, that's elifesciences.org, or you can subscribe on iTunes. This is a Naked Scientists production. I'm Chris Smith, and you can find out more about what we do on our website at nakedscientists.com. I shall be back, meanwhile, with another look inside eLife next month. Until then, goodbye.
The eLife podcast from eLife, the open access journal for outstanding research in the life and biomedical sciences. Online at elifesciences.org.